All right, good morning, everybody. Welcome to day 2 of the IKCS. I hope everybody had a wonderful first day, a wonderful evening in Nashville last night. We're going to get started with our hometown hero. Brian Reaney is going to take us through the Academy of Kidney Cancer Investigators and talk about some of the DoD grants. So, Brian, welcome to the stage.
All right, thanks everyone. I'm not sure I've ever been called a hometown hero before, so thank you, Phil. So we've presented at this conference before. I have the pleasure of being the dean of what's called the Academy of Kidney Cancer Investigators. And what this is the DoD grant program, which you're all familiar with. I guess it was about 4 years ago now, introduced this mechanism of having a virtual mentoring academy. And so we've had 2 or 3 early career investigators admitted every year into the academy, and I'm just going to take you through in 5 minutes sort of what it is, and then we're going to hear from the 3 early career investigators from this year. We have 1 in person and actually 2 virtual, but we'll hear from all 3 today. That's our logo. I love it. So this is our structure. I have the pleasure of being the dean. Jim Brugarelis is the chair of my advisory board, and then you see, I know Laurence and Monty are here. This has tended to be more basic science and basic translational, so we've sort of modified our advisory board over the years for more lab-based translational investigators, and you can see them there, and they're all wonderful, and they basically donate their time. They have, a very small amount of salary support that they get, but they're really doing this out of the goodness of their heart. And then just quickly, we're now at 4 years, so basically when the early career investigators apply, they apply with a specific project, and you'll hear about that, and then they apply with a mentor, and it's a 4-year grant for them. It's a 5-year grant for the academy, it's a 4-year grant for them, so we're finally sort of full as this year, as we've admitted the 4th year. This is year 1. I'm not going to read all this to you, but just to sort of show you the different folks, and many of those folks are at this meeting. And it's really, I think over the years, what I've noticed is that there's much more interaction among the academy members. Obviously, when there was just 3 in the first year, it was less. But as we've grown and as they've matured as investigators, we're seeing a lot more sort of cross-pollination and collaboration, which is, I think, the whole idea of this academy. So this was our class last year, and then this is who you're going to hear from today. Maxine Sun from Dana-Farber with a cardiovascular-related, Nirban who's here in the audience, who's going to present on his work on SEMA-B5, and then Chen, who I think will actually be the first presentation. So the presentations today will be from the bottom to the top of the slide, virtual, in-person, and virtual. And then this is just from a recent, we set out goals when I wrote the initial grant of what do we want to accomplish here, and we tried to sort of quantify them and say, let's quantify, abstracts and publications and everything you see listed on the left here, and those cumulative achievements over the first 3 years are what we put together, I think, pretty impressively. It's a really impressive group, and again, I think the collaboration and the cross-pollination has sort of grown over the years. We have a didactic series that we do not quite monthly, maybe it's 6 to 8 times a year, and many of you in the room have done it. I just saw Brian's done one, so this is a little bit of a dated slide, but we do these again about once a month or so, and again, people around the country and around the world have been very generous with their time to come on for 45 minutes and talk about kidney cancer, and it's really a great way to expose sort of that early career investigator to really almost like a one-on-one session. I mean, there's just them and the speaker, and there might be some of the advisory board folks and mentors on, but it's maybe a dozen people, and it tends to be a really nice and rich interaction. So we're proud of that, and that will continue. And in the last year, we have a website, right? Nobody without a website. So this is just a place where everything that we are sort of gathered in one place, right? It talks, basically the slide sort of that I've presented is gathered in one place. It gives little bios of the mentees and their projects. We have a link to our videos here. It's sort of everything in one place. So check it out. And it also has a link to apply. So I think one of the goals of the academy obviously is to be self-sustaining and get as many applicants as possible. So we just passed a cycle for DoD submissions. But it's a really great way to get funding for early career investigators. The idea is to draw them into kidney cancer and then obviously to keep them there, right? And I think it's been successful so far. All right, so that's it for my brief intro. Our first presentation I think is from Chen Yao, and it's virtual. Chen is expecting a baby any day. That's why she's not here. It's a pretty good excuse. So I don't know if we'll just play it from the back of the room. I'm looking towards the back of the room.
It's a great pleasure to speak at this International Kidney Cancer Symposium. I really wish I could be present. However, my OB told me that my baby can come at any time, so it's better to be on the safe side. Today, I will talk about my project on targeting stem-like CT cells immunotherapy against kidney cancer. I'll start with a review that we write recently. So there are mainly three classes of drugs for kidney cancer. One is small molecule drugs that directly inhibit tumor growth. There's also tyrosine kinase inhibitors that inhibit the angiogenesis. And lastly, there's immunotherapy that aim to invigorate exhausted T cells to eradicate tumor cells. Even though immune checkpoint blockade has revolutionized the treatment for renal cell carcinoma, we know that around 70% of kidney cancer patients still progress despite the immunotherapy treatment. CD T cells become exhausted during cancer due to constant T cell stimulation and the immunosuppressive microenvironment. Exhausted T cells have high expression of co-inhibitor molecules like PD-1, TIM3, CTLA-4. They also have reduced effector function and proliferative potential. However, one question that lost is, if all CD T cells are exhausted during cancer, how can patients remain a long-term immunity? In the last few years, people have found that anti-tumor T cells are actually heterogeneous. There's the stem-like T cells, which have high expression of transcript factor TCF1 encoded by TCF7 and have a low expression of co-stimulatory molecule TIM3. These stem-like T cells can maintain the long-term anti-tumor immunity by self-renew and differentiate into other T cell subset. More importantly, people have found that these stem-like T cells are important for the efficacy of immunotherapies, including vaccination and checkpoint blockade. Using mouse models of tumor, people found that if you deplete the stem-like T cells, the efficacy of immunotherapy is decreased. So the stem-like T cells is critical for the efficacy of immunotherapies. Given that the immune checkpoint inhibitors relies on the expansion of stem-like T cells rather than the reversal of T cell exhaustion, enhancing anti-tumor immune response of the stem-like T cells may improve the efficacy of ICI during kidney cancer. In the last few years, my lab has been studying the transcriptional and epigenetic regulation of stem-like T cells. We found that transcript factors, including TOX and BAC2, are important for T cell stemness and the long-term immune response. We also elucidated the transcriptional and epigenetic regulation underlying the resistance of stem-like T cells against exhaustion. However, the molecular program governing the efficacy of ICIs in kidney cancer while harnessing stem-like T cells is under development. So in this proposal, I hypothesize that the therapeutic efficacy of ICIs against renal cell carcinoma is determined by the quantity, metabolic fitness, as well as tumor microenvironment of the stem-like T cells. So, given the critical role of stem-like T cells in anti-tumor immunity induced by ICIs, the limited number of tumor-specific stem-like T cells in renal cell carcinoma may be a bottleneck for ICI treatment. Our preliminary data show that a subunit vaccine, including a toll-like receptor ligand and an anti-CD40 agonist antibody as adjuvants, induce a robust immune response by stem-like T cells. So we hypothesize that the efficacy of PD-1 blockade against RCC is determined by the magnitude of anti-tumor response of stem-like T cells. So in aim 1.1, we will use a synergistic murine RCC model to evaluate whether enhancing the anti-tumor CD T cells by the therapeutic vaccination can improve the efficacy of PD-1 blockade against RCC. Even though we know that stem-like T cells were also found in human RCC, their antigen specificity and the prognostic value in PD-1 blockade are unclear. So leveraging the sample of renal cell carcinoma patients treated with ICIs at UT Southwestern, and also machine learning algorithm developed here, which will predict the TCR peptide and MHC interaction, we hope to define the antigen specificity, differentiation, as well as the quantity of tumor-specific stem-like T cells in renal cell carcinoma patients. More importantly, we'll determine whether the abundance and transcriptome of anti-tumor stem-like T cells can predict the clinical outcome of PD-1 treatment in RCC patients. So we know that for T cells response to immunotherapy, they need sufficient metabolic support and also immunostimulation from antigen-presenting cells. However, renal cell carcinoma forms a hypoxic tumor microenvironment, limiting the oxygen supplies to T cells and consumes large amount of glucose, which is nutrients needed by anti-tumor CD T cells. Our preliminary data show that stem-like T cells relies on greater oxidative phosphorylation than terminally exhausted CD8 T cells. In addition, renal cell carcinoma is infiltrated by immunosuppressive myeloid cells. Thus, the unmet metabolic demand and the immunosuppressive tumor microenvironment of the stem-like T cells may limit the efficacy of ICI. Tyrosine kinase, which inhibit androgenesis and reformat the vasculature of CD8 cells, has been effective in treating RCC and shows synergy between with ICIs. However, how does TKI directly regulate anti-tumor T cells, especially stem-like T cells, is unknown. So our preliminary data suggest that TKI treatment alter the expression of genes in metabolic pathway in tumor-infiltrating T cells, and also reshape the landscape of tumor-infiltrating myeloid lineages. So in MA2, we hypothesize that TKI treatment will improve the metabolic fitness as well as tumor microenvironment of stem-like T cells in RCC, which will enhance the efficacy of immune check blockade. So in aim 2.1, we will use single-cell metabolism assay to determine how TKI modulate the metabolism of anti-tumor CD T cell subset, especially stem-like T cells, in a murine model of RCC with or without PD-1 blockade. And in our endpoint 2.2, we'll evaluate how the intra-tumor niche of stem-like T cells in mouse and human RCC is affected by TKI monotherapy and the TKI plus ICI combination therapy. So my hope is that by successful accomplishing this study, we will reveal molecular and cellular mechanism by which the stem-like T cell mediates the immune response against RCC, and based on those findings, we can find novel strategies that will improve the anti-tumor efficacy of ICI through combination therapies. With that, I would like to thank my mentor, Dr. Celeste Simon from UPenn and Jim Broglos from UT Southwestern, also people in my lab who have generated the majority of primary data, and my collaborators, Twitchy and Rolf at IKCS Western, also my funding resources.
Forgot if you're supposed to clap after virtual presentations or not. All right. So next we have an in-person presentation. Nir Kundu is here to talk about the role of SEMA5B in kidney cancer. Nir, thanks for joining.
Good morning all. This is Anirban Kundu, and I'm a junior faculty in the University of Alabama at Birmingham, and today I'm going to present very preliminary data on the project on SEMA5B's role in kidney cancer. And actually, I'm thankful to DoD for funding this project, and I will continue this project further in my new lab in the University of Arizona Tucson that I'm going to starting off in January 2024. Now, actually, first I will try to see the actual history of the project, that how we actually ended up with SEMA5B, and then I will talk about some brief of SEMA5B's role in kidney cancer, and then finally I will talk about the mechanism that is in progress. Because this project, it was started as our aim to actually look at the epigenetic dysregulation in kidney cancer, and we found that PRDM16 is a tumor suppressor in kidney cancer that was epigenetically silenced in kidney cancer, and by a gain of function analysis, we found PRDM16 can make a complex with the transcription repressor CTBP, and that's how this repressive complex can silence the expression of HIF-regulated genes like SEMA5B. Actually, SEMA5B is a member of semaphorin family proteins. They are shown on the left. And there are actually several types of semaphorin molecules. They are generally present on the cell membrane, and they can interact with its receptor molecules. They are neuropilins or plexins, and that's how they generate the signaling. These semaphorin plexin signaling, they have role in several biological processes, and the dysregulation of this signaling has been implicated in many diseases, and including cancer. And when we looked into the kidney cancer, then we found that SEMA5B transcript is highly upregulated in kidney cancer tissues compared to the normal. That is shown in left, and on the right it shows the patient-matched normal tumor samples, and it shows the SEMA5B protein is highly up in the tumor samples. And I can say from best of my knowledge that this is the first actually SEMA5B protein data in the literature so far. And in fact, when we check the pan-cancer model, and we found that particularly in the KIRK database, that is the kidney cancer database, only the SEMA5B is highly up as compared to the other tissues or actually other tumors. And in the GEO database, and we find that SEMA5B is in fact very high level actually expression in the metastatic renal cancer tissues. And it suggests that SEMA5B can have a role in the metastasis. As SEMA5B is highly up in kidney cancer, so first we wanted to look at that whether SEMA5B is HIF-VHL regulated actually or not. And on the left, actually we show that by VHL gain of function and by HIF stabilization, we found that SEMA5B is a HIF targeted gene. And after silencing the SEMA5B by shRNA, and we can even silence the tumor phenotypes both in vitro and in vivo. Now coming to the mechanism part that is in progress, on the top left, it shows the general notion of SEMA14 molecule. They are either cell membrane like class 4 to 7, or the class 3. It is the extracellular SEMA14. So we try to see that whether SEMA14 5B is present on the cell membrane or not. So we first have the RCC cells, and then we specifically label the cell membrane protein using biotin, and then we actually pull down the proteins by using streptavidin beads, and we found that the bound fraction that represent the extracellular or the cell membrane protein, and that unbound fraction, it represents the intracellular proteins. And we found in both the cell lines that SEMA5B is coming actually with the other intracellular proteins like tubulin and 1433, and it actually also shows their intracellular EGFR molecule, and that in the bound fraction it shows only the EGFR, that is the bona fide cell membrane protein. So actually this suggests that SEMA5B is an intracellular protein, and this is opposing the notion that SEMAs are present intracellularly. And then we analyze the SEMA5B protein deduced from different transcripts of SEMA14 of the SEMA14 5B, and we found that the proteins from most of the transcripts of SEMA5B, they do not have the signal sequence that can export SEMA5B on the cell membrane. So this further supports our finding that SEMA5B can be an intracellular protein in kidney cancer. So the take-home message is that from my work, that SEMA5B is a novel HIF target oncogene, and as SEMA5B's expression is highly up in kidney cancer, so we can specifically target this SEMA14 axis to specifically target the kidney cancer cells. But there are a lot of work to be done, and I hope that I will pursue this direction in a positive way. And finally, I want to acknowledge my mentors and my lab members and Urological Care Foundation grant that supported my background work for this project, and finally DoD for funding my future works. Thank you.
Thanks, Sunir. Come sit up here. Come on. All right, thank you for that. Appreciate it. And our last presentation, which will also be virtual of this session, is from Maxine Sun from Dana-Farber.
My name is Maxine, and I am a research scientist in GU Oncology at the Dana-Farber Cancer Institute in Boston. And today I will be presenting to you on the effect of clonal hematopoiesis on cardiovascular outcomes in patients diagnosed with kidney cancer. So we will be talking about what is clonal hematopoiesis, and specifically go over what are mosaic chromosomal alterations. And then I will briefly present to you the findings that I have been able to uncover with respect to mosaic chromosomal alterations in patients diagnosed with kidney cancer, which I believe are highly relevant. So, clonal hematopoiesis is a known biomarker in hematological cancer for its development and progression. In 2014, there was a study where investigators reported that clonal hematopoiesis was associated with a 40% higher risk of all-cause mortality. And that, of course, wasn't surprising, but what was surprising and unexpected about that study was that when they dug a little bit deeper, they found that the cause of death driving that increased risk of all-cause mortality wasn't actually caused by hematological cancer, but it was mostly driven by incident coronary heart disease as well as ischemic stroke. So, what is clonal hematopoiesis? And this, as a general definition, is as people age, their tissues accumulate an increasing number of somatic mutations, most of which are inconsequential. Occasionally, there will be a mutation that will arise and it will confer this fitness advantage on a cell. When this process happens in the hematopoietic system, a large proportion of circulating blood cells end up derived from this single mutated stem cell. And this particular outgrowth is what is termed as clonal hematopoiesis. So, right after that unexpected discovery, where they found that clonal hematopoiesis has implications in atherosclerosis, there were other studies that were able to pinpoint implications of clonal hematopoiesis in inflammation. And so, in this experimental study, it was shown that loss of TET2 increased both the production of the precursor form interleukin 1 beta, as well as augmented the activity of NLRP3 inflammasome complex, which then increases the conversion of the pro interleukin 1 beta into active interleukin 1 beta. And we have also previously shown in a study of about 1,800 individuals who were recruited at the Montreal Heart Institute Biobank that carriers of clonal hematopoiesis of indeterminate potential, or otherwise known as CHIP, had significantly higher high sensitivity C reactive protein concentrations than non carriers, C reactive protein being an established biomarker of inflammation and atherosclerosis. So, what are mosaic chromosomal alterations? So, as a general rule, clonal hematopoiesis can first occur in single nucleotide variants, SNVs, as well as indels, in driver genes that have been typically associated with myeloid malignancies known as CHIP mutations or clonal hematopoiesis of indeterminate potential. Alternatively, you also have mosaic chromosome alterations or MCAs, and these are SNP array detected large scale copy number of events that have been also shown to drive the phenomenon of clonal hematopoiesis in its own distinct mechanism. What is meant by mosaicism here is simply that there is a mixture of normal and mutant cells. And during the course of our life, we inherit the DNA that we inherit from our parents, and then our cells throughout life acquire somatic mutations. And in most of these cases, these cells with the mutations die. In other cases, they do end up proliferating. And the most extreme example of that is the phenomenon of a tumor, but most of the time it's not that extreme. Chromosomal alterations is basically when there is a significant change in the content of a cell, and these changes can be classified as a loss, a gain, or when a whole chunk is entirely replaced by another. This is called copy number neutral loss of heterozygosity. Numerous health consequences have previously been associated with MCAs. MCAs include the mosaic loss of the Y chromosome, and the health consequences include cardiovascular events in the general population. And so in our study, we thought that it would be interesting to assess the impact of MCAs on cardiovascular related outcomes, but focusing specifically on patients diagnosed with cancer. And so we did this using the UK Biobank, and we had about 50,000 individuals diagnosed with cancer. And our hypothesis stated that carriers of MCAs would have a higher risk of cardiovascular related death and all cause death compared to non-carriers. And so indeed, that's what we found in our multivariable Cox regression models. We did observe that carriers of MCAs had a significantly higher risk of death from coronary artery disease than non-carriers, as well as a higher risk of death from cancer-related causes and any cause mortality. What was particularly intriguing about that finding was that as we did sub-analysis focusing on this association of MCA and cardiovascular outcomes per cancer type, we found that for patients diagnosed with kidney cancer, so that was about 1,900 individuals in a UK Biobank, carriers of MCAs had a significantly higher risk of death from cardiovascular disease, as well as a higher risk of death from coronary artery disease compared to non-MCA carriers. And what was notable about this observation was that MCAs was actually not significantly associated with kidney cancer related mortality. So this really strengthens the robustness of MCAs being a specific marker for cardiovascular related outcomes. And so based on these preliminary findings, we thought that it would be worthwhile to investigate the influence of clonal hematopoiesis on cardiovascular outcomes, focusing on patients diagnosed with kidney cancer, but using cohorts with more detailed information, including disease characteristics as well as the treatments that they have received. And so our first proposed aim in this project was to assess shared heritability between kidney cancer and hematopoietic related phenotypes, including CHIP, MCAs, loss of the Y chromosome, etc. And then the second aim essentially tries to replicate the effect that we found of MCAs and cardiovascular outcomes in the UK Biobank. So we're going to propose looking at associations of clonal hematopoiesis, whether it being CHIP or MCAs on cardiovascular outcomes in patients diagnosed with kidney cancer using institutional data such as the Dana-Farber Cancer Institute cohort and likely bigger population-based cohorts with genomic data. So in order to if we're able to replicate the effect across these different cohorts, then it really strengthens the robustness of this marker as well. And finally, for the last aim, because clonal hematopoiesis has inflammatory profile that has very significant implications in inflammation, we decided to compare the inflammatory gene profiles and expression of carriers of clonal hematopoiesis and non-carriers with the possibility of identifying specifically what inflammatory pathways are involved. And so the totality of these proposed aims fortunately resulted in a positive answer for the Early Career Investigator Award of the DoD. And I am very grateful as a result to be part of the Academy Kidney Cancer Investigator Group. So to summarize, cardiovascular health is becoming increasingly important for patients diagnosed with kidney cancer as we are increasing the number of therapies that are available in the first lines and subsequent lines, clonal hematopoiesis appears to have an influence on the risk of cardiovascular outcomes in patients diagnosed with kidney cancer. So there is a definite interest in trying to investigate this role furthermore, and also to try to understand and characterize the specific biological pathways that this potential association is stemming from. So thank you very much for your time today, and I hope to be seeing you soon.
Helps if I turn it on. Thanks everyone for your attention. That wraps it up for this session. I just want to thank KCA for giving us the platform to present. You can see there's a lot of different and interesting work, and it's fun for me to be a part of it. So I think we're going to move on to our next section, which is molecular diagnostics and biomarkers. I think Yusuf Zakari is leading this. I can't really see anything because it's so bright out there. I see Brian and Alan and others, so here we go. Thank you.